Hello to all of you who are joining us today, and thank you. My name is Michelle Klock. I am delighted to welcome you to today's webinar titled Single Cell Analysis, Unveiling Tumor Heterogeneity, which will be presented by Dr. Rainer Blaesius, an R&D manager with BD Biosciences. This webinar is being co-sponsored by BD Biosciences and the Current Protocols Journals of John Wiley & Sons Publishing. Now, I'd like to introduce today's speaker. Dr. Blaesius received his Ph.D. in biochemistry from the Free University in Berlin. After continuing his graduate work on G-protein coupled receptor signaling in his postdoctoral studies at UNC Chapel Hill, he developed GPCR-based assays for drug discovery as a senior scientist at Caro Bio USA. Upon joining BD Technologies, he led teams in the development of antibodies for cancer screening and staging assays, as well as a novel screening platform for stem cell media. In his current position as R&D manager, Dr. Blaesius, with his colleagues, has pioneered the application of flow cytometry for solid tumor tissue analysis and fluorescence-activated cell sorting, or BD-FACS, to enable surface marker characterization and single-cell molecular analysis for solid cancers. Throughout today's webinar, I would like to encourage you to submit any questions by clicking on the Ask a Question box at the bottom of your screen. Your questions will not be seen by any of the other attendees, and the Q&A session will take place after the presentations. The webinar will be recorded and available for viewing in the next few days, and we will send you an email with details of how to access the on-demand webinar. So, with a very warm welcome, please take it away, Dr. Blaesius.
Hello, everyone, and welcome to today's webinar. Thank you to Michelle for the kind introduction, and as Michelle said, my name is Rainer Blaesius. I work as an R&D manager at BD Technologies and Innovation, and this is located in North Carolina and is the corporate R&D center for BD. I will be talking today about how to use surface markers and flow analysis and sorting and downstream gene expression studies in order to discover cancer cell properties which are hidden from bulk analysis. To begin with, we might ask the question, why do we want to do single-cell analysis for cancer research at all? From a purely conceptual standpoint, the fact that biopsies are made up of tens of millions to upwards of tens of billions of cells would probably tell us that they're likely not all alike. But in addition, pathologists who have stared at tissues on slides for more than 100 years have always observed many different cell types in this tissue. Molecular methods, on the other hand, were mostly designed to detect genetic abnormalities, mutations and such, but for the most part neglected the fact that there was a variety of cells. And more recently, especially over the last 1 to 2 decades, the fact that solid tumors are actually an ecosystem of stromal cells, epithelial cells, immune cells, and so on, in addition to the cancer cells, has become much better appreciated. And the cancer cells themselves in most tumors show considerable variety. The reason this is becoming more important is that the previously limited view failed to fully explain the cancer properties, and it's now a widely accepted view that moving to this ecosystem view will lead to a much improved understanding and ultimately to better patient outcomes. Today's webinar will be based mostly on data that was generated in patient-derived xenograft or PDX models. These models are generated by implanting human tissue into immune-compromised mice, and they grow tumors which, for the most part, resemble the patient tissue in many different ways. There are some limitations, and the absence of a functional immune system is probably the most important one, but despite that, these models have proven to be very useful to study actual tumor tissue. So I'm starting with a very simplified mock-up of a tissue slide here, simply to prove a point important for today's webinar. So as we look at this mock-up, we realize there's a lot of variety in it here, a lot of different colors and different objects, and one typically might need some help to identify cancer cells in a tissue slice. And expert eyes are capable of identifying the tumor cells here by shapes and sizes and so on. But still, as we think about what the current status of molecular analysis is what happens next in almost all cases is the following. So the first step in a molecular analysis very often is that we throw the cells into a blender and we make a homogeneous mix of it. And if we look at the outcome of that, what we generally find is that we're looking at the mix of all cells and as if they were a homogeneous mass, and the readout at the bottom here is actually not very easily readable. And in contrast to that, if we are able to sort the cells beforehand, as you see, the point I'm trying to make here is the result becomes a lot more readable. Now, taking this to a somewhat more scientific frame of mind, I'm showing a figure here which is from a very widely cited review article by Hanahan and Weinberg, which shows the variety of cells in a tumor lesion, and it shows the different types of cancer cells as well as fibroblasts, endothelial cells, pericytes, and so on. So suffice it to say there is a whole architecture here, lots of different cells, and one of the first questions might be, aside from their shape and size, what else do we have to identify these different cells? One of the most useful ways to identify these cells are surface markers, and I'm showing a range of surface markers here for each cell group. And I want to state that these are shown as CD markers, which are very common to immunologists and hematologists. But you will also discover that most or all cell groups are marked by multiple markers, as well as some of the markers appear in different groups. And I'm explaining that in order to make the statement that most cell groups will need to be identified with multiple markers. So it's a combination code, if you will. So then if you're not an immunologist or a hematologist, you might be unfamiliar with the CD nomenclature, but you can think of it simply as something akin of a zip code. It's simply an unambiguous surface marker nomenclature, and you might actually be much more familiar with these surface markers than you thought if you just hear the CD name. And all I want to say with that is these are familiar molecules, antibodies exist to them, and they're highly useful in tissue cells. In fact, we were, when we started this, kind of surprised because the CD markers have been developed, like I said, in immunology and hematology, but their use in solid tissue is something that turns out to be highly useful. And as I mentioned earlier, what the data I'm about to show is from tissue derived from patient-derived xenografts, and in that case, the surrounding cells, other than the cancer cells, are mouse cells, so stroma and endothelial cells, and et cetera, are mouse cells, and only the cancer cells are human cells. And I just want to say that because in the following data slides, all I'm going to report about is human cells, and the mouse cells will be excluded from the data display. So taking us back to my tissue mock-up from before, what this means is that the mouse cells surrounding the tumor literally drop out of the picture. And what we're left with are just the human cancer cells. But within within this community of neoplastic cells, you find quite a variety, as I try to indicate with the different colors here. This is something that was already recognized more than 40 years ago, and I try to highlight this here by mentioning a publication by Gloria Hepner and colleagues who in the late 70s already tried to submit a paper on this heterogeneity, and it was rejected by the reviewers, citing as reason that everyone knew that cancers are monoclonal. And she was successful to overcome this by stating the trivial fact for developmental biologists that she herself originally was monoclonal but became undeniably heterogeneous. And this is truly something that in cancer looks like it has been forgotten for many years, but lately this is becoming much more appreciated, that it's a very important property of cancer lesions. I'm showing the fact that this acceptance is really ongoing even in the clinical field by just mentioning a very nice publication that appeared last year by Carlo O'Malley and colleagues who have been suggesting ecology and evolutionary indices in order to deal with this variety and with the dynamic of tumor cells themselves. And suffice it to state that intratumor heterogeneity and the evolution of cancer cells have become an important and widely accepted notion. So with this in mind, the question comes up technically, how do we deal with this variety? And what I'm showing in this slide is a workflow which we have established here at BD, which shows solid tumor flow analysis and sorting. And it starts on the upper left with a tissue slice. Generally, what we work with is around 100 to 1,000 milligrams. We have worked with needle biopsies as well, but the restriction in tissue mass there, of course, makes it a little bit more challenging. First, this is being taken through a dissociation step, which employs mechanical and enzymatic means. We have developed a reagent that's called tumor dissociation reagent. It is available from BD currently as a special order product. The cells are then taken through a filtration step where aggregates are taken out, and downstream process from there is basically just like what some of you might know from blood-based cells. It includes cell washes, red blood cell removal, and followed by staining and flow analysis. And if you do flow analysis, everyone familiar with flow knows that you can also do flow sorting. And what's shown at the bottom here are downstream modalities after you have your sorted cells. You can do quantification, you can do DNA sequencing, gene expression, single-cell gene expression. So the boxes marked in green here are methods where we have proof of concept already established. And of course, once you have sorted cells, you can subject them to all other kinds of modalities as well. Before I'm fully going into the actual data part of the presentation, I'm going to explain how the flow cytometric data is presented today, because we have over 1,000 plots at the base of the data I'm showing today, so of course it would not be useful to show those, but I'm showing these in a summarized format. So on the upper left here you see a plot, and the flow cytometrist makes the gating decision and determines what's the percentage of cells positive for the marker in question. This percentage then is entered into the chart on the right. And I want to emphasize the fact that percent means percent of human cells. So again, keep in mind the tumor tissue comes from a tumor-bearing mouse, and there are mouse cells present, but the mouse cells are excluded from the subsequent analysis. So all you're going to see is percent of human cells. And at the bottom you see the profile that we generate with this. And you see multiple dots here. Each dot represents a single mouse, and this is all from the same model. And when I refer to a model, it means that one model is generated from one patient. So different models, different patients. One model can have replicates of different mice, and that's shown here. And word to the markers that were chosen at the bottom here. So these are 10 markers which we chose from literature. They had been used as presumed cancer stem cell markers or for similar reasons for solid tumors before. But I also want to say that while I'm showing data almost exclusively for these 10 markers, our more recent data indicates that there are many other markers that you can use for these purposes. So we have done this with a range of breast cancer samples, or breast cancer cohorts, I should say. And if you look from top to bottom here, it becomes very clear that mice within one cohort always exhibit highly similar surface marker profiles. So the replication within the cohort is very high. A second point that becomes clear here is that each cohort seems to have a distinct profile. So what's generally referred to as intertumor heterogeneity is being picked up by the surface marker profiling. And on top of that, you will see that markers frequently are expressed on some cells, but not all. So like in BRC13, if you look in the middle there, CD133, there would be an expression of 60 or 70% or something like that. And what this means is simply that not all cells in this tumor express this particular marker. In other words, we're seeing intra-tumor heterogeneity. Furthermore, some of the cohorts we analyzed came from the same breast cancer model. So for anyone unfamiliar with PDX models, I should add that you can propagate the tumor tissue through mice multiple times. That means you can take the tumor tissue grown from the first mouse, implant it into the next mouse, use that material, implant it into the third mouse, and so forth. This is generally referred as passaging, and passage numbers usually go up to maybe as high as 10. On this slide, you see examples of tumors at different passage numbers. So our analysis indicates that a surface marker profile is a stable property along the growth of the tumor. I should mention here that all mice were obtained from Jackson Labs, so it's a commercial source, and unfortunately we did not have access to the clinical material, so we cannot show based on our data that this extends to the clinical samples, but I would argue that this is a worthwhile thing to do in the future. So we can take the same data and express this in form of a heat map. And again, I want to stress the fact the data you saw on the previous slide is the same data as this. But as you can see on the color scale on the right side here from 0 to 100, so this again is the percentages for each sample positive for the particular marker. And on the left side of the slide, you can see color coding for each cohort, and what becomes apparent immediately is that so there's an automatic cluster analysis that the mice or the samples from each cohort cluster together. And as I mentioned earlier, some of these cohorts were actually from the same model, and that's shown here, and with just a few exceptions, all of these cohorts from the same model also congregate together. In other words, the surface marker profile is capable of sorting almost every mouse to the respective model. As we have seen from the data so far, these surface marker profiles are reproducible, they're distinct for each cancer model, and they suggest intratumor heterogeneity. So a question that comes up is, can we identify distinct subpopulations? And the way that we can address that question is through FACS sorting of the human cells, and we can follow that up by focusing on specific subpopulations within those human cells. So the marker that we have chosen to test this hypothesis that we can actually distinguish different types of cells, in this case, is CD184. This was a choice because it's one of the markers that has been reported as a potential cancer stem cell marker. It is also increasingly an important drug target, but it was also chosen because in this particular cohort, we clearly saw a distinction between CD184 positive and CD184 negative cells. So this slide shows you on the left the population in a flow plot together here. So first we distinguish between human cells and mouse cells. The mouse cells are identified by a cocktail of species-specific antibodies. And then in the next plot, you see the human cells alone, which then are stained with CD184, and you see the distinction between 184 positive and 184 negative cells. So those populations are split, and on the right you see the plots verifying inflow that we really have a pure positive or a pure negative population, respectively, for downstream analysis. So you see four groups of cells here: unsorted, human only, CD184 high, and CD184. We repeated this with 3 different mice, so it's a triplicate, a biological triplicate, and it's all from the same model, i.e. from the same single patient. So we subjected these samples to whole transcriptome RNA-seq, and first we looked, and we got excellent assistance here by Joel Parker from University of North Carolina in Chapel Hill. And we looked first at a PCA, sort of a global analysis to ask the question, are these cells different from each other in their gene expression profile or not? And you can see on the y-axis here a difference between the unsorted cells and pretty much everything else. And on the x-axis, you see a difference between 184 low and 184 high cells. And in the plot here, you see that the replicates all segregate together, so it really looks like a true biological signal that we're looking at. The next step was a comparison of the gene expression profile of our samples with the breast cancer samples in the Cancer Genome Atlas, or TCGA. The heat map on the right shows expression levels of the various genes across the y-axis of the plot, and along the x-axis you have all the samples in the breast cancer samples in TCGA. Above the heat map, you see a color coding bar there, which indicates the molecular subtypes you see in the legend on the upper left of the heat map. These subtypes were pioneered by the lab of Chuck Peru at UNC Chapel Hill, and Joel Parker is affiliated with that lab. His analysis determined that all of our samples, so the unsorted as well as the subpopulations, group within the basal subtype. It's not necessarily surprising to find the unsorted cells there because it's a triple negative mouse model, but the sorted populations, of course, are quite distinct from each other. So according to this analysis, at least they are not any more similar to one of the other molecular subtypes than they are to the basal breast cancer. So they are all bona fide basal breast cancer cells. Then the following step of the gene expression profile analysis is to ask the question, which of these genes between the CD184 high and the CD184 low population are different? And we discovered at a very high stringency, around 770 genes, which are were differentially expressed between 184 high and 184 low. And the genes actually fall into very distinct buckets. The CD184 high cells express genes specific for proliferation and cell cycle. In contrast to that, the CD184 low cells have higher expression of genes connected to hypoxia, EMT, ECM regulation, basal markers, and so on. So what becomes clear here is these two different subpopulations seem to have distinct roles within the tumor lesion. This impression was amplified by subsequent pathway analysis, which again showed that, and I want to stress with high statistical significance, that the CD184 high population seems to be engaged in proliferation and pretty much nothing else. In contrast to that, the CD184 low population is engaged in various environmental regulatory activities like extracellular matrix, angiogenesis, epithelial cell differentiation, and so on. This interpretation is consistent with observations by other investigators who have described cells with different proliferation speed, and generally the slower cells are being regarded as having a bigger role in drug resistance. They're usually candidates for cancer stem cells, and I want to also mention the fact here that the cancer stem cell model has undergone a maturation, so I might prefer using the word tumor-initiating cells because it's not as a static model. But suffice it to say, these CD184 low cells might be the more interesting population. And right around the time that we did this, a publication by Levi Garraway's lab appeared. And this paper describes a gene signature which is called the axel gene panel, which originates actually from an earlier lung cancer paper by Zhang et al., but in the Levi-Garraway publication a couple of years ago, it was used to investigate melanoma, and they described a specific set of 100 genes. And we tested the presence of these 100 genes in the CD184 low cells, and discovered that they were significantly higher expressed in this population. Again, this echoes this impression that the CD184 low cells within this breast cancer lesion are tied to possibly to drug resistance and to organizing of how these cells grow. Most of the data as shown today was published in Molecular Cancer Research last year. You see the key figures of the paper at the top here. You saw that we've further expanded the profiling to further models, and the heat map of the previous statuses on the upper left here. It has convinced us that this is a very worthwhile approach altogether. And more recently, we've actually expanded this to more markers, and we've realized that there are actually many more markers that you can utilize for this approach. So altogether, we believe that by doing so, it's going to become even more powerful. In summary, our data indicates that surface marker profiles are reproducible, that they're distinct between each model, presumably between patients as well, and that they're suitable to demonstrate inter- and intra-tumor heterogeneity. And this intra-tumor heterogeneity, reflective of the ecosystems I mentioned initially, is suitably addressed by FACS-sorting subpopulations. Our finding that these subpopulations are very distinct and biologically coherent, have biologically coherent gene expression profiles is further proof that the surface markers are actually reflective of meaningful phenotypic differences. So taken together, this observation tells us that our methods really provide unparalleled insight into the tumor ecosystems in contrast to the currently still common bulk analysis. So at the end, I show the workflow again that we've put together, and I hope that I was able to convince you that this addresses tumor heterogeneity. And again, on the left side, we start with a tissue slice. We can dissociate this with the help of the dissociation buffer that we have developed here. Then you can analyze it with an analyzer, and most of the data that you saw was generated with an LSRX20. The sorting experiment were done with a FACS ARIA, but we also offer the FACS Melody, which is easy to use. And on the far right, I'm showing the Rhapsody platform, which is a recent addition to the BD portfolio, which allows you to do single-cell analysis. I want to stress the fact that the data that you saw was not generated with that, but as I was explaining earlier, once you have your sorted cell populations, you can subject them to any downstream modality that you want, and I'm hoping to be showing data generated with a single-cell gene expression soon. Finally, I would like to acknowledge all the people who did the work and supported the project. Mitch, Friedrich, Warren, Eileen, and Francis are the people here in North Carolina who have basically generated all the data. Stacy Chu assisted in statistical analysis. The Research Animal Facility here in North Carolina provided fantastic services. Shannon and John supported the project in the leadership team, which we're very grateful for. And on the other side here, on the right side, from the West Coast, Smita and Aaron have been instrumental in developing the methods that were used in these studies. And Kevin, Linda, Marissa, and Skip are the leadership members in California who have supported the project. And also, as I mentioned before, Joel Parker from UNC has provided expert assistance in analyzing the gene expression data. And lastly, I would like to thank all of you listeners, and I hope you found this webinar enlightening, and I would be very pleased to take any questions that you might have.
Thank you very much for that wonderful presentation, Dr. Blaesius. Now let's go ahead to the question and answer segment. So if you haven't yet submitted a question for our speaker, now is the time to do so. So our first question is going to be, so Dr. Blaesius, do you consider CD148 low cells to be stronger in the role against drug resistance?
So based on the data, I would venture to guess yes, but I have to emphasize that we don't have functional data, but that would be my hypothesis, yes.
Oh, great. We have another question. Oh, sorry, go ahead.
For this particular model, I might add.
Perfect. So another one of our other audience members was wondering why PDX models? Why not directly from human primary tumors just after surgery?
We had two reasons for that. One is that we really wanted to hone our technology and show the reproducibility. And of course, with patient tissue, you don't have that. And second is a practical issue. We don't have access to tissue on site here that's easy to schedule and where you get sufficient material. So this was a pragmatic choice as well.
Great. So we have another question. One of our speakers is wondering, could you please talk a little bit about the protocol used for dissociation, specifically with regards to cellular debris and getting a clean cell solution with high viability?
Sure. I might start at the end here. So viability, do keep in mind that a lot of tumors actually have necrotic cells. So to expect very high viability with a tumor of any appreciable size may be unrealistic. So one has to account for that. But with that in mind, we have seen viability from some tumors around 90%. So we feel pretty confident that per se our dissociation protocol does not limit viability too much. And as far as the protocol itself is concerned, it is rather straightforward the way that I described it. So you basically use, depending a little bit on your preference, scissors, scalpels, or razor blades, along with the tumor reagent, the dissociation reagent that we developed. Debris is something that, of course, you will end up with at the lower size, but you account for that during the gating process in the flow cytometry.
Great. We have another speaker or another audience member thought this was a very interesting approach, especially for pathologists, and would like to know how it would compare with direct NGS analysis.
Direct NGS analysis, well, I'm not exactly sure I understand the question completely, because direct NGS analysis, the way I understand it, is you go for DNA sequence. So what we were after is gene expression of particular groups of cells, because if you look at the bulk of the cells, you get the average of gene expression profile from everything in there. And as I was trying to point out earlier, and I'm sure you're aware, there is a great variety of cells, and if we separate them first, then the gene expression profile with regards to what it says about function is much more accurate to the actual cell group. So I'm not 100% sure I really answered that question, but that's the best, the way I understand it.
Well, we'll take it. Thank you. So would you expect differences in tumor heterogeneity from PDX models if human cancer cells were also implanted with human stromal cells or other types, rather than using mouse cells to establish the tumor architecture? A bit long.
But that's a really good question, and it speaks to the general comparability between PDX-derived tissue and clinical samples. I would expect, especially in some cases, to find differences there that directly are a cause of the human environment versus the mouse environment. But I think there's enough experience in the whole field with PDX samples in general to show that there is heterogeneity present, and obviously our results would argue that way as well, so that at least some of the mechanisms are there, and it's easy to investigate those in PDX models for the reasons that I stated, but clearly these methods were
generated in order to now take them to clinical samples, and that is actually also what we're doing. It's just that we don't have the amount of data that I was showing today. That's almost impossible to generate with similar resources with clinical samples.
Great. So for our next question, how do you compare between PDX and circulating tumor cell-derived mouse models?
Also a really good question. We have not tackled that in any comprehensive way. So I think that would be something very worthwhile to undertake, but unfortunately I don't have any data in that regard.
Great. So for our next question, what do I want to do? So if you do single cell sequencing, do you need to bulk cell RNA sequencing? I'm not sure if that's a good question.
Well, let me just say this. The single cell sequencing is based on barcoding. So you do get data from each single cell, but the sequencing reaction itself is done in aggregates, which also makes this a whole lot cheaper than if you had to run a sequencing reaction per each cell. I'm hoping that addresses at least part of the question.
Yeah, that one was a little tricky. So another question regarding the PDX model. So what is the profile of cells used for transplantation, and do you think it properly represents the original human tumor heterogeneity, especially if comparing single cells from the original tumor to the PDX transplanted cells?
Yes, again, that's a really good question. Now, unfortunately, all the models here were commercial models, so we obtained them from Jackson Labs, and we did not have access to the patient material. My guess would be that you do find at least a partial representation of the patterns that we identified. But again, I base this on the fact that all the literature that I know about that compares the human samples and the derived PDX models, very frequently there's similarity there. And because the surface marker pattern is so characteristic, my guess would be that you find those, but we have not done that experiment yet. I'd be very eager to do it.
That's great. So do you have any experience with live and dead cell ratios before and after freezing down patient samples?
Yes, also a really good question, very technical. So you can do this in various ways, and I just skip the ways that don't work as well. The best way to do this in our hands is to dissociate the cells first, freeze them down in the presence of DMSO, basically like a regular cell culture freezing conditions. And the viability, you do take somewhat of a hit, and it depends a little bit so I can't give firm percent numbers, but let's say you start with a high viability sample, 90%, you actually get maybe even up to 80% viability out. If you start lower, you also get lower, but we have successfully done this repeatedly with samples that started lower. So it's clearly feasible, but you do take somewhat of a viability loss.
Great. Thank you so much. For our next question, so the data presented here was based on solid-derived cancers. Would this approach also work on liquid-derived cancers, for example, AML?
I'm not exactly sure what, in terms of the heterogeneity, absolutely, but of course the dissociation aspect then does not apply. But yes, I mean, and there is quite a bit of work that has been done for heterogeneity in blood-borne cancers as well.
Great. We have an interesting question. Can this technology also be used for microorganisms?
Oh boy, I'm the wrong person within BD to ask.
Yeah, that one's kind of a wild card.
Right. I do know that flow cytometers, in principle, yes, that can be used. But again, in my hands, we have not used that. And I would refer you to some people at BD Biosciences that can try to connect you with a person that might have experience there.
Gotcha. Yeah. Microbiology is near and dear to my heart. So another question, are you concerned about the dissociation process with antigen or RNA integrity?
It's a good question, and in general, yes, one should be concerned about the quality of one's analyte. But in our hands, this actually has worked quite reliable. With regards to RNA integrity, we have not seen any loss compared to, let's call it the gold standard, to just take the tissue piece and extract bulk RNA. And with regard, we have compared changes directly, and they're very minimal. With regards to integrity of the antigens, we have analyzed that and have actually done somewhat of a survey. There are a few markers that are susceptible to changes, and we basically try to take that into account, possibly choose other markers. But the great majority, I would say greater than 90% of the markers, don't change at all in this process. There's a publication that I would refer you to by Laurie Ailes in Toronto. She has done a survey, I want to say, of around 300 different markers under different dissociation conditions. That's also a really good indicator, and she also found that the great majority of markers are actually not affected. So we're pretty confident that with regards to marker integrity, we don't take much of a loss.
Oh, great. So we have another technical question from one of our audience members. Do you lyse red blood cells before phenotyping? And the reason they were asking is because lysis changes the scatter profile of the cells, and they were wondering if you have any suggestions.
We do lyse the red blood cells because it is the regular workflow, like with blood-derived cells, and we actually have not really looked at the changes there. So it's since we haven't focused on the morphology either, but just on the marker profile, we haven't really taken that into account.
Okay, great. Do you think that this is applicable for the immune cells in clinical samples?
Yes. So the methods clearly were developed with that as the ultimate goal. And like I said earlier, for mostly practical reasons, we chose PDX samples because they're reproducible and accessible to us. But we have some experience with clinical samples there, and they all indicate that, yes, our methods are actually very suitable for clinical samples. And we will be publishing some of that in the future. Great.
We have one question. I'm not sure if this is something you can answer. One of our audience members was wondering about what the current status of single-cell proteomics is with relation to this technology.
Well, all I could do there is basically to refer you to the literature there, because I don't really have on hand or hands-on comparison. It's kind of hard. Now, what I would say is that for anybody who has access to a flow cytometer, or even if you currently don't, I was mentioning some of the cytometers that are now being produced for that are easier to use. But if you do have access in any form, then it's something that is based on a lot of experience. And other means of proteomics, mass spec, for example, they tend to be a little more involved technically in terms of setup and everything. So I would just provide that as an incentive to go this route. But of course, there are other ways to do this as well.
Great. So I think that might be it for our questions, unless anybody has any last-minute questions they would like to submit through the question and answer session. I'll give you a couple of seconds to type anything out that you might want to ask Dr. Blaesius while we have him. All right, I'm not seeing anything else pop up. So with that, I'd like to go ahead and wrap up. So we're wrapping up a few minutes early. So thank you so much to everyone for all of those excellent questions and for joining us I want to remind you that today's webinar has been recorded and will be available for viewing within the next few days. For those of you who registered for the talk, we will be sending you an email with details on how to access the recorded webinar on demand. So on behalf of today's speaker and our sponsor, BD Biosciences, we sincerely appreciate your attending today's webinar, and we look forward to your attendance at future webinars by Wiley and Current Protocols. So thank you so much.